Right. Good morning, everyone. Welcome to Biomed Journal Club. I'm your minister of postdoc in this lab. And today I'm going to talk about this bioarchive paper titled TwinCell Large Causal Cell Model for Reliable and Predictable Therapeutic Target Prioritization. Yeah, let's start with reading this abstract. Right. You're finished reading. So, how do you think?
Need general capsule.
Yeah. So, right. So, for me, the first sentence seems like kind of gap sentence, but it looks like more background. Yeah. So it did not cover mm, some detailed previous approaches to solve this issue. And, yeah, but anyway, everyone would agree with that this problem is very difficult, and so every attempt to solve this issue is anything. The later part is also more what it can do rather than how it works. Of course, it explains some brief idea, but yeah, it didn't explain some true idea. So, is there any other comments about this session? Then let's move on. All right. So, if there's such a cells, it would each cell would have its own cellular state. But if we apply some perturbation here, it changes the cellular state of a cell, and this perturbation is like a drop, chemical perturbation, or some critical gene knockout things. So state change means, for example, like this. So for the state A, so maybe without any certain intervention, the single-cell sequencing data looks like this, but after the perturbation, the data will change like this. But the key thing you need to be sure, you need to keep in mind is there is no one-to-one correspondence between the pre-cell state and post-cell state, because if we do single-cell sequencing, and the cell dies. So this state change is, maybe you can think that distribution change. Anyway, so this kind of perturbation, single cell data is many researchers are focusing. So there are some public data set that hold the single cell data under some perturbation conditions. So for example, this paper is published in Nature Methods. They collect 44 publicly available single cell perturbation response to this data sets. And another data set, hold 100 million. So this is, I think this is quite popular, and some well-known institute for developing virtual cell called ARC Institute makes this data set, and they are training some AI virtual cell models based on this data. So yeah, it contains 100 million single cell data, and it incorporates more than 1,000 types of small molecular perturbations. And this data is in vitro data set, so they made a cell line of cancer cells and apply the perturbation and measure the synthesis sequencing data. So with this data, previous studies mainly focus on predicting the post-perturbation cellular state. So for example, in 2019, I think people think this is the first attempt of using AI model to predict this cellular perturbation. So here, different colors represent different cell types, and they apply the same perturbation to these different cell types. So this circular means pre-perturbation, and triangle means after perturbation. So, and the aim of this model is predicting what would be the after perturbation state of not trained cell type based on the trained cell type. So to do this, they use the autoencoder type AI model. So they just embedded the single cell data into latent space, and they assume that the same perturbation had the same vector change. So with this idea, they predict this perturbation for unseen this new cell type. And later, so I think you guys remember this paper. So I think Aksa presented this model in the Journal Club, and this model uses the diffusion model to predict the perturbation factor. But they uses the single cell data, and also some embedding of the drug information. So they predict this cellular state change due to this perturbation. And another model is called state. So this model was developed by the ARC Institute, and it also uses the table 100 million data. And so I think it uses some transformers architecture, but maybe in my next Journal Club, I will cover this related details. So, yeah, as I said, previous studies mainly focus on predicting the cellular state change. So it's more like forward problem. But the TwinCell, the main target of today's Journal Club, predict not the cellular state change, but they uses pre and post state and predicts what was the target or key pathway affected by this perturbation. So it is more like backward problem. So do you get the idea? Yeah. So the key idea of this TwinCell is using the already known whole cellular molecular interactions. So we contain the protein, transcription factors, and genes. So you can think that this network is a very big network of. So it includes every interactions already known. Of course, this could be incorrect, but they assume that this interaction is the true. So the TwinCell does not infer a new interactions, but it relies on just some prior knowledge of the network, so the network structure. And so with this network, what TwinCell does is it first get the pre-cell and post-cell state, which is the single cell data, and it uses some foundation model for single cell data called GenFormer. And so it embeds this data into the latent space. So some details of the GenFormer is in just briefly explained in the appendix of this slide. But simply saying, so you know that some transformer takes the token. So large language model takes words as a token. So instead of the word, this GenFormer gets the token of genes. So each gene gets a token, but its order changes depending on the expression level. So high rank genes, high expressed gene has a higher rank, and lower expressed gene is lower rank, and it gets the order and it takes the input as a big. It takes the order tokens to get this latent vector. Then with this latent vector, the TwinCell predicts these node weights. So for example, like these values. But I noticed that in the paper, it did not specify why the architecture of TwinCell. So we don't know how it works. But anyway, it gets these two inputs and predicts whole is node weights. Then this node weight has a value between 0 and 1. And the meaning would be like this. So for example, if we think this node 1 and node 2, this value is represent some kind of probability of node 2, condition on node 1, and these two cell states. Maybe you can think in this way. So this represents the probability that when node 1 is perturbed, it is caused by node 2. Yeah, so TwinCell is trained to predict this probability. Yeah, do you get the idea? Then, after getting this node weights, it again goes back to this pre-cell and post-cell states, and it analyze what is the differentially expressed genes, which means that so there's pre-cell and post-cell, and for each gene, you can analyze whether the gene expression distribution of the pre and post is statistically different, like this. So it only collects the set of genes that differentially express. Then maybe we can take these differentially expressed genes in the network. Then for this DG, we are going to find the targets of that cause these differentially expressed genes. So the target candidates would be these proteins. So for example, it's P1 to P4. Then for each proteins, we calculate the probability that P1 affected to DG, which would be the multiplication of this three values. So, and we can do this for this whole, the target candidates, for example, like this. So P2 would be, the probability would be like 0.16, and P3 is summation of these two pathways. So it'd be like this. And the P4, there's no pathway to DG, so probably would be 0. And based on this probability, so you can think this is like a score to identify the target, and maybe based on this result, you can conclude that, okay, the main target was the P3. Then, yeah, actually, that's all. That's all about the TwinCell. It's very simple way. And also, after the TwinCell is trained to maximize the probability of the already known target protein. And after we get the main target of this this perturbation, then maybe we can think various pathways like this. So for example, for this DG and this target, we can get two pathways. Then, so for example, the pathway 1 and pathway 2. Oh, and again, we can calculate the probability of each pathway. Then maybe we can conclude it like this. So most all other pathway was pathway 2. So this is how the TwinCell also predict the causal pathway from this data. So yeah. So this TwinCell can predict what was the target of the certain perturbation and already known DGs. Then we need to evaluate its performance compared to previous models. So to do that, what we can, so they develop some benchmark framework called TwinBench. What does is, so like the TwinCell, it gets the pre and post cellular state data and predict the target score, which is the probability we've seen before. And based on, so there are so many, so target candidates, and each target candidates have their own score, right? And based on this score, it can rank the target and it can normalize. So it's just like, so if there are n number of candidates and the true target rank is R, then normalized rank is between 0 to 1. 0 means higher rank. 1 means the most low rank. And also, previous approaches usually set some threshold like K rank. And if the target is within K, then it evaluate, oh, this model is okay. But they didn't like this. So they mainly focus on this normalized rank, and also it evaluates this. So maybe it's evaluating something like the precision. So if certain target affect to most of the perturbation, then maybe this model predicts that this target is the cause of every perturbations. Then we don't want that situation. So that means if wrong input comes to wrong or some irrelevant data about this perturbation comes in, then the model should not predict the true target. So to evaluate this, it randomly permitted the cellular gene, for example, gene label. So that means it disrupted the input data, then calculate the target score again. Then maybe it can calculate the probability of the known true target, and so it repeat this process many times and can construct this null distribution. And if the true target score from the upper part is statistically significantly high, then we can say that, okay, this model is good. So again, so it is evaluating like this. The target score should decrease under some disruptive inputs. Yeah. Do you get this? Okay. So this normalized rank and p-value is the two main values to be used to evaluate this model. But another thing we need to consider is the kind of p-value threshold. Maybe we can take the 0.05, but it uses more clever way. So for example, so we evaluate this model for several perturbations. And each perturbation, it would have some true target. Then for this true target, we can calculate this normalized rank and p-value. Then we can make it maybe draw this kind of the scatter plot. So you can think each corresponds to its target, and this value represents the p-value and this normalized rank. Then these dots here has low p-value and low normalized rank, so which means the high rank. So this is good result, but this is, even the rank is high, very high, the p-value is low, because that means, so this is has some population bias. So this means for very many perturbations, this target is kind of common cause. So it's not specified to this perturbation. So we don't want this. And so maybe we need to quantify the performance from this scatter plot. So to do that, we set some threshold of this p-value. For example, 0.05. And by changing this threshold, we can calculate something called recall and IMMR. So recall here is the fraction of true target has low p-value. The low p-value than this threshold. Then of course, if we increase the threshold, then the curve will increase. And at the same time, this means that with low threshold, this will take high value when the prediction is okay. The predicted true target has low p-value, and IMNR is, so we can collect all these dots lower than this threshold and take the mean value of this normalized rank. This should be low. So it takes 1 minus of this mean normalized rank value, so that it gets high value when all targets with low p-value have low normalized ranks. So recall means that this dot should be located in here, in this line, and this means that this value should locate in here. Yeah. So, and both measures are important, right? So that they calculate the F1 score, which is the harmonic mean of these two values. Then we can draw like this graph. So you have two graph of recall and IMNR. We take the harmonic mean of these two graphs, then we'll get this graph. Then with this low threshold, if this value is quite high, then we can think that this model performance is quite nice. So it calculates the area under curve of this F1 curve. So this higher area under curve represent higher performance. And this framework, we can interpret in this way. So good model, it means true target should have low normalized rank. And also the target score should decrease when gene labels are randomized. Yeah, so this AUC F1 score would be the main quantification of this performance. And yeah, this framework was developed by these authors. Yeah. So they evaluated various models, including TwinCell, to the in vitro data. So zero-shot cell line means that there are several cell lines and divided into two groups, so trained cell lines and test cell lines. And they evaluate its performance in the test cell line. And you can see that this AUCF1 score is high, only for this TwinCell. And if you draw these three curves like this, you can see that the other models, the AUC is quite low, but it has quite high the F1 AUC score. And here, the other models is one was the state I explained before, and the other one is I think it's quite too naive approaches. So actually, there are not many models predicting the target rather than the cellular state change. So they brought some thing called network medicine. So it is also in the appendix. The paper about this was published in 2016 in Asia Communications, and the approach was like this. So if the target is closer to some disease network, then we think that this is the target protein. So in this paper, we know the DEGs, right? We thought this model calculates the distance from the certain target to the DEG network. Then it uses as some target score, and based on this target score, it predicts the main target. Then of course it has very low performance. And another which is just simple regression. So actually we have some kind of each cell, we have a vector, right? Pre and post. We do some dimension reduction, but not kind of very special dimension reduction, but it only selects a highly variable genes, or it uses the gene former encoder. Then it concatenates and do the linear regression to predict the target score. Very, very naive approach. So again, this has very low performance like this. But interesting thing is state is not good compared to these naive approaches. Maybe this is due to it is out of distribution cell line. Yeah. And yeah, another thing is, so previously they tested in the in vitro data, and now we do in vivo analysis, more related to some clinical things. So they considered the data about this SLE, some disease. So in Korean it was like this, and it is some chronic autoimmune diseases, which means that the immune cells attacks our body. So they collected data, especially the B cell data. And oh yeah, another thing is this interferon alpha receptor pathway is known for the main cause of these diseases. So they collected B cells of the normal and the disease state, and they applied the TwinCell here, and the TwinCell predicted some key known pathways like this. So this was the interferon alpha receptor pathway. So this is quite good result because TwinCell identified the true critical pathway, even it was trained only in vitro cell data. But of course, this pathway was not the rank 1. Actually, it was rank 11. There were several more targets and pathways. So I'm pretty sure this model is not perfect. And so it also tested these various models seen before to various clinical sets here. So for each disease, actually there are several known targets, and they set some known target as a train set and others as a test set. And they train this TwinCell model and check whether it indeed correctly identify these targets. So maybe I think the train and test data set is not so many, but yeah, anyway, it's saying that yeah, their model works quite well. Yeah. In summary, this TwinCell model uses the known some cell molecule interaction network, and also it uses some the foundation model gene former and unknown architecture. It predicts the node weights, and eventually it predicts the target of this model, target of certain perturbation, and also the causal paths. Yeah, that's all.